long before cities, farms, and written history. Homo sapiens began moving into a world that was already inhabited by other humans. Small groups traveled beyond Africa and entered the vast landscapes of Eurasia. They followed coastlines, crossed river valleys, moved through open grasslands, and eventually reached regions shaped by severe winters and rapidly changing climates. They carried fire, stone tools, clothing made from animal hides, and knowledge passed between generations. They were intelligent, cooperative, and highly adaptable. But they were not perfectly prepared for everything that waited ahead. The animals were unfamiliar. Seasons followed different patterns. Winter could transform a productive valley into a frozen landscape where food became difficult to find. Sunlight weakened in the northern months, and temperatures fell far below those experienced by many earlier generations. There were also dangers that could not be seen. New environments contained unfamiliar bacteria, viruses, parasites, and other microorganisms. A population entering these regions for the first time did not possess generations of local biological experience. Its immune system had been shaped by a different history, but Eurasia was not empty. Neanderthals had already lived across Europe and Western Asia for hundreds of thousands of years. Their communities had endured repeated shifts between colder and warmer periods. They knew the movement of local animals, the locations of stone and water, and the ways in which shelter and fire could make a deadly winter survivable. Their bodies had also been shaped by that world. Neanderthals were strongly built, with physical proportions that helped conserve heat. They made carefully designed tools, hunted large animals, controlled fire, and cared for injured members of their communities. They were not the crude creatures once imagined in outdated popular stories. They were another human population with its own long evolutionary history. Farther east lived people whom scientists now call Denisovans. Their known physical remains are extremely limited, but their DNA reveals a much larger story. Denisovan populations were genetically diverse and occupied parts of Asia. Different groups of Denisovans later contributed ancestry to different modern human populations. For a time, several human populations shared the planet. They had separated from one another long before, developing distinct genetic histories, yet the separation was not complete. When Homo sapiens met Neanderthals and Denisovans, some of those encounters produced children. The evidence remains inside living people. Many modern populations outside Africa carry small fragments of Neanderthal-derived DNA. Denisovan ancestry is especially substantial in some populations of Oceania and is also found in varying amounts across parts of Asia. These fragments do not form a complete Neanderthal or Denisovan genome inside any modern person. They are scattered sections of DNA that have been divided and rearranged through thousands of generations. Most of the original archaic ancestry did not survive. Some fragments disappeared by chance. Others were gradually removed because they worked poorly within a mainly Homo sapiens genetic background or reduced the likelihood of successful reproduction. but certain fragments remained. Some may have survived simply because they caused no major disadvantage. A smaller number appear to have become more common because they helped their carriers respond to particular environments. This process is known as adaptive introgression. The idea is simple. When a population enters a new environment, useful genetic changes can arise through mutation and slowly spread through natural selection. But that process may require many generations. There is another path. A population can acquire genetic variants from people who have already lived in that environment for a very long time. Evolution does not always have to create the same solution again. Sometimes it borrows an existing one. Neanderthals and Denisovans had faced Eurasian climates, pathogens, sunlight, and landscapes long before the arrival of many Homo sapiens groups. Through interbreeding, some of their genetic variation entered the newcomers. If an inherited variant helped a person survive infection, tolerate environmental stress, or produce healthy children, that fragment could become more common among later generations. This does not mean that Homo sapiens survived only because of archaic DNA. Culture was essential. People made clothing, developed new tools, shared food, maintained social networks, and altered their behavior far more quickly than genes could change. Human flexibility allowed communities to survive in deserts, forests, coasts, mountains, and frozen plains. But culture and biology worked together. Our ancestors did not enter Eurasia as a finished species perfectly adapted to every new challenge. They continued to change after they arrived. Some of that change came from natural selection, acting on their own variation. Some came from new ways of living, and some came from encounters with other humans. Neanderthals and Denisovans eventually disappeared as distinct populations. Their communities, languages, and traditions did not survive into the present. Yet their disappearance was not complete. Some of the people who vanished became ancestors of the people who remained. Their inherited fragments would later influence how certain human populations responded to infection, sunlight, altitude, pain, and other biological pressures. Some of those variants may have helped our ancestors survive a dangerous ancient world, but the world did not remain ancient. The same biological responses would eventually operate inside cities, hospitals, heated homes, and bodies exposed to conditions their original carriers could never have imagined. The genes remained. Their meaning began to change. The meetings between these populations were not a single event. Homo sapiens did not cross one border, encounter one Neanderthal group, and then continue eastward unchanged. Human expansion happened over thousands of years, through many landscapes and among many communities. Some groups may have seen one another only from a distance. Others probably competed for hunting grounds, water, caves, or access to stone. There may also have been peaceful contact, shared camps, exchanges of food, and the movement of individuals between communities. Genetics cannot reconstruct every encounter. It cannot tell us what language people spoke, whether a meeting began with fear or curiosity, or how two strangers learned to trust one another, but it can reveal that some encounters produced children. One of the clearest examples comes from Denisova Cave in the Altai Mountains of Siberia. Scientists examined a small bone fragment from a girl who lived tens of thousands of years ago. Her DNA showed that her mother was closely related to Neanderthals, while her father belonged to the Denisovan population. She was not a distant descendant of an ancient mixture. She had one Neanderthal parent and one Denisovan parent. Her existence proves that separate human populations sometimes met directly and formed families. It also suggests that such contact was not biologically impossible, even after long periods of separation. The genetic story of modern people shows that mixing occurred more than once. Different populations outside Africa carry different patterns of Neanderthal ancestry. These patterns cannot be explained by one simple meeting involving one small group. Some Homo sapiens populations encountered Neanderthals in Western Eurasia. Later migrations and additional contacts appear to have added new fragments of ancestry in other regions. The Denisovan story is even more complex. Modern populations in Papua New Guinea and other parts of Oceania carry substantial Denisovan ancestry. Some populations in East and Southeast Asia carry smaller, but genetically distinct Denisovan contributions. This indicates that the people we call Denisovans were not one uniform population living only near a single Siberian cave. They included several groups that had separated from one another and adapted to different parts of Asia. Each encounter left a different genetic trace. At first, a child with one parent from each population would have inherited large blocks of DNA from both sides. But DNA does not remain arranged in the same way forever. During the production of eggs and sperm, chromosomes exchange sections in a process called recombination. Large inherited blocks are broken into smaller pieces. Those pieces are passed to some descendants, but not to others. With every generation, the original ancestry becomes increasingly divided. After thousands of generations, archaic DNA survives as scattered fragments distributed across the genomes of living people. This is why two people can each carry Neanderthal ancestry, but possess different Neanderthal-derived segments. One person may carry an ancient variant related to immune function. Another may carry one affecting skin biology. A third may carry neither of those, but retain different fragments elsewhere in the genome. No modern person carries every surviving Neanderthal variant, but across millions of people, many separate pieces of that ancient genetic history remain. The commonly mentioned percentage of Neanderthal ancestry can therefore be misleading. For many people with substantial ancestry outside Africa, the average is often described as roughly 1 to 2%. That does not mean a person had one recent Neanderthal ancestor who contributed exactly that amount. The mixture occurred far deeper in the past. The surviving percentage is the result of repeated recombination, population movements, natural selection, and random inheritance over an immense number of generations. Some fragments became rare simply because not every descendant inherited them. Others disappeared because they reduced fertility or worked poorly with the rest of the Homo sapiens genome, and some remained because they were neutral or useful. When a beneficial fragment increased in frequency, it could spread far beyond the small community in which the original encounter occurred. A variant inherited from one Neanderthal individual might eventually appear in many thousands of descendants. Later migrations could carry it across entire regions. This helps explain why the biological effect of an ancient meeting could continue long after the people involved had died. The encounter itself may have lasted only a moment. Its genetic consequences could last for tens of thousands of years, yet ancient ancestry did not spread evenly. Population size, migration, geographic isolation, and later mixing all changed the pattern. Some modern groups inherited more Denisovan ancestry than others. Some retained particular Neanderthal fragments that became rare elsewhere. Even populations within Africa can carry small amounts of Neanderthal-related ancestry, largely because groups with Eurasian ancestry later migrated back into Africa and mixed with local populations. Human history has never been a simple movement in one direction. People left regions, returned to them, separated, reunited, and exchanged ancestry again. For this reason, the evolution of modern humans is better imagined as a network than as a tree with permanently isolated branches. Branches divided, but sometimes they touched again. Genes crossed between populations, even when cultures and communities remained distinct. A fragment that originated in one human group could survive inside another, while the population that first carried it disappeared. This mixture did not erase the differences between Homo sapiens, Neanderthals and Denisovans. They remained populations with separate evolutionary histories, but their boundaries were not absolute. Our species emerged from Africa with its own biological inheritance. In Eurasia, that inheritance encountered other human lineages and absorbed small parts of them. Most of those borrowed fragments faded. Some remained quietly in the genome. And when our ancestors faced unfamiliar infections, climates, and landscapes, a few of those fragments would begin to matter. The most immediate threats in a new environment were not always the ones our ancestors could see. A predator could be avoided, a storm could be anticipated, a fire could provide warmth against the cold, but bacteria, viruses, and parasites were invisible. When Homo sapiens entered Eurasia, they encountered microorganisms that had circulated among local animals and human populations for countless generations. Their immune systems had evolved elsewhere and carried a different history of exposure. Neanderthals had lived in these regions for hundreds of thousands of years. During that time, natural selection had acted repeatedly on their defenses against infection. Variants that improved survival during epidemics or reduced the damage caused by local pathogens could become more common. When some of those variants entered Homo sapiens through interbreeding, they offered a possible shortcut. The newcomers did not need to wait for every useful mutation to arise independently. They could inherit parts of an immune system already shaped by Eurasian conditions. One important example involves a group of genes known as toll-like receptors. These receptors form part of the innate immune system, the body's rapid first line of defense. They recognize common molecular patterns associated with microbes and help trigger inflammation before the slower, more specialized immune response is fully activated. Among the best studied are TLR1, TLR6, and TLR10. These genes sit close together in the human genome, and some modern populations carry versions of this region that appear to have entered Homo sapiens from archaic humans. The inherited variants can alter how strongly immune cells respond when they detect possible infection. In an ancient environment, faster recognition could make the difference between containing a pathogen early and allowing it to spread through the body. A person whose immune response activated quickly might survive an infection that proved fatal to someone whose defenses reacted more slowly. That advantage would not need to be dramatic. Even a modest improvement in survival or reproductive success, repeated across many generations, could increase the frequency of the inherited region. But a sensitive warning system creates another problem. It may react when the danger is small, or when there is no true danger at all. Some archaic variants in the TLR region have been associated with stronger immune activity and increased susceptibility to allergic conditions. The same biological tendency that may once have improved the detection of microbes can, in a different environment, contribute to unnecessary inflammation. Content reference OAS8EUE Index DO. This does not mean that Neanderthal DNA directly causes a person's allergies. Allergic disease is influenced by many genes, childhood exposure, air quality, diet, infections, medications, and other environmental conditions. An archaic variant may slightly change the probability or intensity of a response. It does not determine the outcome by itself. The distinction is important. Evolution does not usually leave behind simple genes for single diseases. It leaves biological systems filled with compromises. The immune system must be aggressive enough to destroy harmful organisms, but restrained enough to avoid damaging healthy tissue. Too little inflammation allows infection to spread. Too much inflammation can injure the body it is meant to protect. There is no perfect setting that works equally well in every place and every century. Another important archaic contribution lies in the OAS region. OAS1, OAS2, and OAS3 help defend the body against viruses. When cells detect certain forms of viral RNA, proteins produced by these genes help activate mechanisms that interfere with viral replication and destroy infected genetic material. Modern humans carry Neanderthal-derived versions of this region. Research indicates that archaic variants can influence the expression and activity of immune genes, including responses to viral infection. Some survived at substantial frequencies, suggesting that they may have been useful under particular historical conditions. Content reference ISI to A1 and index to 1, but usefulness depends on the pathogen. A response that works well against one virus may be less effective against another. A strong early reaction may limit infection, Yet excessive activation can also contribute to harmful inflammation. The immune system is not a single weapon. It is a network of alarms, barriers, signaling molecules, specialized cells, and regulatory controls. Changing one part can influence many others. Archaic ancestry may also have affected parts of the adaptive immune system, including regions involved in presenting pieces of pathogens to immune cells. The HLA system is central to this process. HLA molecules display fragments of proteins so that immune cells can distinguish infected or abnormal cells from healthy ones. Human populations possess extraordinary HLA diversity because different variants can recognize different threats. Some researchers have proposed that contact with archaic humans added useful immune variation to modern populations. But this area is genetically complex. Similar immune variants can be ancient, shared across several populations, or inherited through multiple roots. It is therefore difficult to assign every modern HLA type confidently to Neanderthal or Denisovan introgression. The safest conclusion is broader. Interbreeding contributed to the diversity of human immunity, but it was only one part of a much longer evolutionary history. Our immune systems were shaped by ancient pathogens, migration, population decline, diet, animal contact, climate, and repeated epidemics. Archaic DNA entered that changing system and sometimes altered how it responded. The consequences continue today. Modern life has transformed the environment in which these defenses operate. Many people live in heated buildings and drink treated water, use antibiotics, receive vaccines, and encounter a very different mixture of microbes from those faced by ancient hunter-gatherers. At the same time, pollution, processed food, reduced exposure to some microorganisms, chronic stress, and longer lifespans create new pressures. An immune response calibrated for wounds, parasites, contaminated water, and repeated infection may behave differently in a relatively sanitized urban environment. The machinery has not suddenly become defective. The surroundings have changed. This is one reason the phrase, "the price of survival," must be used carefully. The inherited variants were not harmful mistakes, waiting to punish future generations. They were biological possibilities. Under one set of conditions, a stronger inflammatory response could protect a family from infection. Under another, the same tendency might increase the risk of allergies or inflammatory illness. And even today, a variant associated with one disadvantage may still offer protection against a different threat. Natural selection does not plan for hospitals, industrial pollution, or future pandemics. It favors whatever improves reproduction in the conditions that exist at the time. The archaic immune fragments inside modern people are therefore not simply good or bad. They are records of past encounters. They show that when Homo sapiens entered Eurasia, survival depended not only on tools, fire, and cooperation. It also depended on defenses operating beneath the skin. Some of those defenses were inherited from humans who had already faced the pathogens of that world. They helped our ancestors recognize danger more quickly. But an alarm that once sounded at the right moment can continue ringing long after the world around it has changed. As Homo sapiens moved farther into Eurasia, survival depended on more than immunity. The human body also had to respond to cold, dry air, changing sunlight, and landscapes where oxygen itself could become scarce. Skin was one of the first barriers between the body and these new environments. It protected against injury, infection, water loss, and ultraviolet radiation. It also helped regulate temperature and contributed to the production of vitamin D when exposed to sunlight, but sunlight did not remain constant across the world. Near the equator, ultraviolet radiation is strong throughout much of the year. Farther north, it becomes weaker and changes more dramatically with the seasons. This created a complicated biological balance. Darker pigmentation can protect the skin from intense ultraviolet radiation. In regions with weaker sunlight, however, lighter pigmentation can make vitamin D production more efficient. Human skin color did not change through one mutation or one simple evolutionary event. It was shaped by many genes, repeated migrations, local environments, diet, and natural selection acting over long periods of time. Archaic humans contributed some genetic variation to this process, but they did not give modern people a single ready-made northern appearance. Popular images often portray every Neanderthal as pale, skinned, and red-haired. The evidence does not support such a simple picture. Neanderthals lived across an enormous geographic range. Their populations were genetically diverse, and they probably varied in pigmentation just as modern populations do. One Neanderthal version of the MC1R gene has been studied because it may have reduced pigmentation and produced reddish hair. But this archaic variant is not the main cause of red hair in modern people. Most modern red-haired individuals carry different variants of MC1R that arose within Homo sapiens. So red hair is not a visible sign that someone has unusually large amounts of Neanderthal ancestry. The connection is far more limited. Some Neanderthals may have had light skin or reddish hair, but modern pigmentation developed through many separate genetic pathways. Other archaic contributions may have affected the structure and function of skin itself. Several Neanderthal-derived regions are found near genes involved in keratin. Keratin proteins help form the outer layers of skin, hair, and nails. They contribute to physical strength and help protect tissues from the environment. In cold and dry conditions, changes affecting the skin barrier could influence water loss, sensitivity, and resistance to damage. Hair structure may also have affected insulation and protection. Researchers cannot reconstruct the exact appearance produced by every surviving archaic fragment. Many genetic regions influence several traits at once, and their effects may change depending on the rest of the genome. Still, the pattern suggests that skin and hair were important targets of adaptation after Homo sapiens entered Eurasia. The newcomers faced conditions already familiar to Neanderthals. They encountered weaker winter sunlight, colder winds, dry air, and long periods when the body had to conserve heat and protect exposed tissue. Some inherited variants may have provided small advantages in those conditions, But the same rule applies here as it did with immunity. A useful ancient effect does not guarantee an advantage in every modern environment. A skin response that helped reduce water loss in cold air might contribute to irritation under different conditions. A pigmentation variant suited to weak sunlight could increase vulnerability to ultraviolet damage after migration to a sunnier region. Human populations continued moving. Genes adapted to one place were carried into others. Culture also changed the pressures acting on the body. Clothing reduced direct exposure to cold. Shelter limited wind and sunlight. Diet could provide vitamin D when sunlight was weak. Fire altered the amount of time people needed to spend outdoors. Biology did not work alone. Some of the most dramatic evidence of archaic adaptation comes not from skin, but from the mountains of Asia. At high altitude, the atmosphere contains the same proportion of oxygen as it does at sea level, but the lower air pressure means that less oxygen enters the body with each breath. For newcomers, this can cause headaches, exhaustion, poor sleep, and reduced physical performance. Over longer periods, the body often responds by producing more red blood cells and increasing hemoglobin. This can improve oxygen transport, but if the response becomes too strong, the blood becomes unusually thick. The heart must work harder, and circulation can become less efficient. Populations living on the Tibetan plateau developed a different pattern of adaptation. Many Tibetan highlanders maintain relatively moderate hemoglobin levels despite living at extreme altitude. A major part of this adaptation involves a region near the gene EPAS1. EPAS1 helps regulate the body's response to low oxygen. The version common among Tibetan populations appears to have entered the ancestors of modern humans through interbreeding with Denisovans or a closely related archaic population. This is one of the clearest known examples of adaptive introgression, a genetic variant that evolved in an ancient Asian human population later became useful to Homo sapiens living in the same demanding environment. It allowed the body to respond to low oxygen without relying entirely on extreme increases in red blood cells. The effect was not the work of EPAS1 alone. High altitude adaptation also involves other genes, developmental changes, breathing patterns, circulation, and cultural knowledge. People learned where to travel, how to find food, how to build shelter, and how to survive severe cold. But the Denisovan-derived EPAS1 region provided a biological advantage that culture could not fully replace. The discovery changed how scientists understood human adaptation. It showed that modern populations sometimes solved environmental problems by inheriting genetic solutions from other humans. The people who first carried this variant disappeared as a distinct population, yet one part of their adaptation survived in descendants who continued living under thin mountain air. This is the deeper pattern running through human evolution. Homo sapiens did not enter every environment with a perfect biological toolkit. Our ancestors learned, invented, exchanged knowledge, and sometimes inherited useful variants from people who had arrived before them. Skin, hair, pigmentation, and oxygen response were not separate stories. They were parts of the same process. A species moving into unfamiliar landscapes became more capable by combining its own flexibility with fragments of older human experience. Some of those fragments helped the body endure cold, weak sunlight, dry air, and high mountains. They did not create a perfect human form. They simply improved the odds of survival in particular places. And once again, the benefit depended on the environment in which the genes were asked to work. The influence of ancient DNA did not end at the surface of the body. Some inherited variants may also have affected the way people slept, used energy, responded to injury, and experienced pain. These systems seem unrelated, but all of them help determine whether a person could survive in an unpredictable environment. Human biology follows an internal daily rhythm. Light entering the eyes helps synchronize a biological clock in the brain. That clock influences sleep, body temperature, hormone production, appetite, and the timing of many processes inside cells. Near the equator, daylight remains relatively stable throughout the year. Farther north, the difference between summer and winter becomes much greater. Summer days can be extremely long, while winter brings extended darkness. Neanderthals lived under these seasonal conditions for hundreds of thousands of years. Some modern genetic variants inherited from Neanderthals are associated with differences in circadian rhythm and chronotype, including a greater tendency toward earlier activity. This does not allow scientists to reconstruct the exact sleeping habits of Neanderthals. We cannot say that every Neanderthal woke before sunrise or followed one fixed daily schedule. The evidence is more limited. It suggests that some archaic variants influenced biological systems that respond to light and regulate the timing of sleep. In a northern environment, sensitivity to changing daylight may have helped people organize activity during short winter days and take advantage of early morning light. But modern life has transformed that relationship. Artificial lighting allows people to remain awake long after darkness falls. Screens expose the eyes to light late at night. Work schedules often conflict with natural sleep timing. A variant that functioned within a world governed by sunrise, firelight, and seasonal darkness now operates inside homes illuminated at every hour. The gene has not changed. The environment around it has. Circadian rhythms also influence metabolism. The body does not process food, store energy, and release hormones in exactly the same way at every hour. Sleep disruption can affect appetite, blood sugar regulation, and the way energy is used. For ancient humans, energy was never guaranteed. Hunting could fail. Plants disappeared in winter. Illness or injury could prevent a person from gathering food. During periods of abundance, the ability to store energy could improve survival during the next shortage. In cold climates, maintaining body temperature also required considerable energy. Natural selection, therefore, did not favor bodies designed for permanent access to inexpensive, calorie-dense food. It acted in a world where scarcity was common. Researchers have connected some archaic genetic regions with metabolic traits, including the regulation of fat storage, blood sugar, and body composition. But these relationships are complicated. There is no single Neanderthal gene that causes obesity or diabetes. Modern metabolic disease results from interactions among many genes, diet, physical activity, sleep, stress, age, medications, and social conditions. An archaic variant may slightly influence one part of this system. Its effect may be harmless in one environment and disadvantageous in another. A tendency to conserve energy could be useful during repeated famine. The same tendency may contribute to difficulty controlling weight when food is available throughout the year and physical activity is low. The problem is not that ancient genes suddenly became defective. The conditions in which they operate changed more quickly than human biology could follow. The same mismatch appears in the body's response to injury. For a hunter living without surgery, antibiotics, or emergency medicine, uncontrolled bleeding could cause death within minutes. The clotting system, therefore, had to respond rapidly. When a blood vessel was damaged, platelets and proteins worked together to form a clot and limit blood loss. A weak response could be fatal, but a response that is too strong creates another danger. A clot forming inside an undamaged vessel can block blood flow to the lungs, heart, or brain. Some studies have linked archaic ancestry with genetic variants affecting coagulation and cardiovascular risk. The exact effects differ among populations, and no ancient fragment explains an individual case by itself. Still, the evolutionary balance is clear. The body must stop bleeding quickly enough to survive injury, but not so aggressively that it blocks its own circulation. Ancient life placed heavy pressure on the first side of that balance. Modern life often exposes the second. People now live longer, sit for extended periods, travel for hours without moving, and survive conditions that would once have been fatal much earlier in life. A clotting response, shaped in a dangerous physical environment, continues functioning inside bodies, facing very different risks. Pain reveals another evolutionary compromise. Pain is unpleasant because it must force attention. It makes a person pull a hand away from fire, protect an injured limb, or stop using damaged tissue. Without pain, small injuries can become severe before the body recognizes the danger. But pain can also continue after its protective purpose has ended. Nerves may remain unusually sensitive. Signals can become amplified. A useful warning can develop into chronic suffering. One genetic region involved in pain signaling is SCNIA. This gene helps control electrical activity in sensory nerves. Rare mutations in SCNIA can produce dramatic disorders in which people experience either extreme pain or very little pain. Researchers have also identified a Neanderthal derived form of a related genetic region that is associated with somewhat greater pain sensitivity in some modern carriers. The effect is not absolute. A person carrying the variant is not destined to suffer chronic pain, and people without it can experience severe pain for many other reasons. It represents a small shift in probability. In an ancient environment, increased sensitivity may have encouraged faster protection of an injury. A person who noticed tissue damage early might avoid worsening it. But when pain becomes persistent, that same sensitivity can reduce movement, disturb sleep, and damage quality of life. Once again, the biological response cannot be described as simply helpful or harmful. Sleep timing, energy storage, blood clotting, and pain all protected survival in different ways. Each required balance. The body had to wake at useful times without losing necessary rest. It had to store energy without becoming burdened by excess. It had to form clots without blocking healthy vessels. It had to detect injury without turning every signal into lasting pain. Evolution did not create perfect settings for these systems. It produced workable compromises for particular environments. Some of those compromises were shaped partly by genetic variants inherited from other human populations. They helped our ancestors move through cold seasons, survive hunger, recover from wounds, and protect damaged bodies. But natural selection could not prepare those systems for artificial light, permanent food abundance, long periods of inactivity, or lives lasting many decades. Ancient biology continues to solve ancient problems. Sometimes, the problem is no longer there. Thousands of years after Neanderthals disappeared, one of their genetic legacies became visible during a crisis they could never have imagined. In late 2019, a new coronavirus began spreading among modern humans. The virus moved through cities, crossed continents, and eventually reached almost every part of the world. Many infected people developed mild symptoms or none at all. Others became critically ill. Age, existing health conditions, vaccination, medical care, and the amount of virus a person encountered all influenced the outcome. But genetics also played a role. Researchers comparing the genomes of people with different forms of COVID-19 identified a region on chromosome 3 associated with an increased risk of severe disease. The DNA variants in this region were inherited from Neanderthals. The finding did not mean that Neanderthal ancestry made someone certain to become seriously ill. Most people carrying the risk haplotype would never experience the same outcome, and many people without it still develop severe COVID-19. It was one factor among many, yet the discovery was striking. A fragment of DNA that had entered Homo sapiens tens of thousands of years earlier was influencing the response to a virus appearing in the 21st century. The chromosome 3 region contains several genes involved in immune activity and the behavior of cells in the respiratory system. Researchers are still separating the effects of the individual variants within this inherited block. The strongest evidence points toward changes affecting how airway cells respond during infection. In some carriers, the ancient haplotype was associated with a substantially higher probability of respiratory failure or hospitalization after infection with the original forms of the virus. But this raises an important question. Why had natural selection allowed such a dangerous fragment to remain? The answer is that it was not necessarily dangerous in the past. A genetic variant does not possess one permanent meaning. Its effect depends on the pathogen, the environment, the person's other genes, and the conditions in which the immune response occurs. The chromosome 3 haplotype may once have provided protection against a different infection. It may also have remained common through chance, migration, or effects that had little influence on survival before the appearance of SARS-CoV-2. A variant that is harmful during one epidemic can be neutral or useful during another. The COVID-19 story became even more remarkable when researchers examined a different Neanderthal-derived region. On chromosome 12 lies a cluster containing the genes OAS1, OAS2, and OAS3. These genes participate in an early antiviral defense system. When a cell detects certain forms of viral genetic material, OAS proteins help activate an enzyme that breaks down RNA inside the infected cell. This can slow viral replication before the infection spreads further. A Neanderthal-derived version of the chromosome 12 region was associated with a lower risk of becoming critically ill with COVID-19. One ancient fragment increased risk. Another appeared to provide protection. Both came from Neanderthals. Both operated during the same pandemic. This is why archaic DNA cannot be classified as either a gift or a burden. Its effects must be examined variant by variant and threat by threat. The protective region did not make anyone immune to the coronavirus. A carrier could still become infected, transmit the virus, require treatment, or die. The effect was statistical rather than absolute. Across a large population, carriers had somewhat better odds. For one individual, the outcome still depended on many other biological and environmental factors. The contrast between these two regions also reveals the complexity of the immune system. An effective defense requires more than maximum aggression. The body must recognize the virus, limit its replication, recruit immune cells, and control inflammation without causing catastrophic damage to the lungs and other organs. A response can fail because it is too weak. It can also fail because it becomes poorly regulated. The chromosome 12 OAS region acts near the beginning of antiviral defense inside infected cells. The chromosome 3 risk region appears to influence a different part of the response. Their opposite associations are therefore not contradictory. They are reminders that immunity is composed of many interacting systems. During the pandemic, public discussion sometimes simplified these discoveries into claims that Neanderthal genes either caused or prevented severe COVID-19. Neither claim was correct. The inherited variants changed probability. They did not decide fate. They also differed greatly in frequency among modern populations because ancient migration and later population history distributed them unevenly around the world. This pattern could not explain the differences in pandemic outcomes between countries or ethnic groups by itself. Access to healthcare, age structure, occupation, housing, vaccination, public policy, previous health, and exposure all mattered enormously. Genes operated inside those social conditions. They did not replace them. The pandemic offered an unusually clear example of an older evolutionary rule. Natural selection prepares populations for past environments. not future ones. Neanderthal immune variants were shaped by pathogens circulating in prehistoric Eurasia. Some entered Homo sapiens because of interbreeding and survived through thousands of generations. Then a completely new virus appeared. Suddenly, fragments that had traveled silently through human history acquired measurable consequences. One was associated with vulnerability. Another was associated with protection. Neither fragment had evolved in response to SARS-CoV-2. The virus simply exposed differences that were already present. This is the hidden nature of ancient inheritance. Most of the time, a genetic variant remains invisible. A person cannot feel that a small section of one chromosome came from Neanderthals. It does not announce itself in appearance or behavior. Its importance may become clear only when the environment asks the body a specific question. Can the cells slow this virus? Can the immune system control the infection? Can inflammation be activated without becoming destructive? During COVID-19, the answers were influenced partly by encounters that had occurred in the Ice Age. The pandemic was modern. The hospitals, ventilators, vaccines, and genetic sequencing were modern. But some of the biological differences between patients were far older. Inside the response to a new virus, scientists found the remains of an ancient human meeting. Ancient DNA is often discussed as if it were a visible mark. People imagine that a heavy brow, a wide nose, red hair, unusual strength, or a particular personality must reveal Neanderthal ancestry. But archaic inheritance does not work that way. Most surviving fragments are small sections of DNA scattered across the genome. They do not create one recognizable Neanderthal type, and they cannot be identified reliably from a person's face, body, or behavior. A modern person with red hair does not necessarily carry more Neanderthal ancestry than someone with dark hair. A strong jaw, broad shoulders, or a large nose do not prove anything about the amount of archaic DNA in the genome. These features are influenced by many genes and by ordinary variation within Homo sapiens. The same caution applies to personality. There is no reliable genetic test showing that aggression, intelligence, stubbornness, creativity, or social behavior comes from one Neanderthal fragment. Complex traits develop through interactions among thousands of genetic variants, childhood experience, education, health, culture, and environment. Ancient ancestry is not a personality diagnosis. It is also misleading to describe one person as more primitive because they carry more archaic markers. Neanderthals and Denisovans were not incomplete versions of modern humans. They were human populations adapted to their own environments. The small percentage of their DNA found in living people does not place anyone closer to the past or farther from modern humanity. Every living person is fully modern. The percentage itself can also be misunderstood. When a genetic report says that someone carries roughly 1 or 2% Neanderthal-derived DNA, it does not mean that one recent ancestor was 1 or 2% Neanderthal. The mixture occurred tens of thousands of years ago. Since then, recombination has broken the inherited DNA into smaller and smaller pieces. each generation passed on only some of those pieces. The fragments carried by one person are not identical to those carried by another. Across an entire population, modern people collectively preserve more archaic genetic material than any single individual carries alone. But even this collective record represents only a portion of what first entered Homo sapiens. The earliest mixed descendants would have inherited much larger blocks of Neanderthal or Denisovan DNA. Most of those blocks disappeared. Some were lost simply by chance. Human populations were often small, and random inheritance could remove a variant even when it caused no harm. This process is known as genetic drift. Other fragments were reduced by natural selection. A genetic variant that worked well inside a Neanderthal genome did not always function equally well when surrounded mostly by Homo sapiens DNA. Genes interact with one another. A protein produced by one gene may depend on many other proteins, regulatory signals, and developmental pathways. When ancestry from two long, separated populations was combined, some combinations may have reduced fertility, development, or survival. Over generations, those combinations became less common. Scientists have identified regions of the modern genome where Neanderthal-derived DNA is unusually scarce. These areas are sometimes called archaic deserts. Their existence suggests that natural selection removed ancient variants more strongly from some parts of the genome than from others. Reduced Neanderthal ancestry has been observed near certain genes involved in reproduction and in parts of the X chromosome. One possible explanation is that some mixed male descendants experienced reduced fertility. This pattern appears in many examples of hybridization between populations that have been separated for long periods. The groups may still be capable of producing children, but certain genetic combinations can create reproductive disadvantages. The evidence does not mean that every mixed child was unhealthy or infertile. The existence of living descendants proves that many were capable of surviving and reproducing. But even a moderate reduction in fertility could gradually remove particular fragments over thousands of generations. Natural selection also acted differently on different tissues. Archaic variants affecting skin or immunity appear to have been retained more often in some populations, possibly because they provided local advantages. Variants strongly affecting the brain, reproductive system, or early development may have been removed more frequently when they disrupted existing Homo sapiens biology, but the pattern is not simple. A fragment that survives today was not necessarily useful. Some remain because they caused little effect. Some traveled beside a beneficial variant on the same chromosome and increased in frequency with it. Others may have been slightly harmful but too weakly disadvantageous for selection to remove completely. Likewise, a useful fragment could disappear by chance before it had time to spread. The modern genome is not a perfect collection of the best ancient genes. It is the result of usefulness, harm, migration, population decline, recombination, and randomness acting together. This is why commercial ancestry reports must be interpreted carefully. They compare a person's genome with known archaic sequences and identify matching positions. They can reveal genuine pieces of ancient ancestry, but they often present the results through entertaining traits such as fear of heights, back hair, sneezing after eating dark chocolate, or preference for certain sleeping times. These associations are usually small statistical differences. They do not prove that Neanderthals behaved in exactly the same way, and they do not explain an individual person's habits. A matching genetic marker is not a recovered memory from the Ice Age. There are further limits. Scientists possess several high-quality Neanderthal genomes, but only a small number compared with the populations that once existed. Denisovan evidence is even more limited. The DNA available today cannot represent every Neanderthal or Denisovan group that lived across Eurasia. Some modern populations may also carry ancestry from ancient groups whose fossils and genomes have not yet been identified. Researchers sometimes call these ghost populations. Their existence is inferred from unusual patterns in living DNA rather than from a complete ancient skeleton. This means the human past was probably more crowded and interconnected than the names Neanderthal, Denisovan, and Homo sapiens suggest. There may have been other populations, other meetings, and other genetic exchanges that remain invisible. The fragments we recognize are therefore not a complete record. They are the pieces that survived and the pieces that modern science has learned to identify. Most ancient DNA disappeared. Some of what remained helped. Some may have carried a cost. And some survived without having any important effect at all. The genome does not preserve a clean story of progress. It preserves a damaged archive. Large sections are missing. Other sections are difficult to interpret. A few fragments become important only when the environment changes. What remains inside us is not a complete Neanderthal or Denisovan inheritance. It is what thousands of generations failed to erase. Modern science can identify fragments of ancient ancestry with extraordinary precision. Researchers compared DNA recovered from Neanderthal and Denisovan remains with the genomes of living people. They search for sections that are unusually similar and then examine how those sections are distributed among modern populations. This work has transformed the study of human origins. For most of history, the disappearance of Neanderthals seemed complete. Their bones and tools survived, but their biological relationship to living people remained uncertain. Ancient DNA changed that. It revealed that some of the people who disappeared did not vanish without descendants. Their genetic material entered Homo sapiens and continued through later generations. But the surviving record is incomplete. Ancient DNA breaks down over time. Heat, moisture, soil chemistry, and microorganisms damage it. Cold and dry environments often preserve genetic material better, which is one reason northern Eurasia has provided many of the best known ancient genomes. In tropical regions, preservation is far more difficult. This creates a major imbalance in what scientists can see. The human past did not occur mainly in caves with perfect preservation. Entire populations lived in places where their DNA may never survive long enough to be recovered. As a result, the map of ancient human ancestry is still unfinished. Denisovans demonstrate how much can remain hidden. Only a small number of physical remains have been confidently associated with them, yet their genetic influence extends across enormous parts of Asia and Oceania. Modern DNA indicates that Denisovan populations were diverse and separated from one another for long periods. The name describes a much larger human story than the limited fossils initially suggested. Other ancient populations may have left genetic traces without leaving any identified remains at all. Scientists sometimes infer these ghost populations from unusual patterns in modern genomes. Their existence reminds us that the categories used today are practical labels, not complete descriptions of every group that once lived. The ancient world contained communities whose names, languages, and identities are permanently lost. Genetics can show that people met and had children. It cannot tell us what those meetings meant to them. DNA does not preserve a conversation beside a fire. It cannot reveal whether two groups exchanged food, copied tools, formed alliances, or fought over territory. It cannot recover the feelings of a parent raising a child with ancestry from two populations. The genome records inheritance, but not memory. This distinction matters when we say that Neanderthals or Denisovans survived in us. They did not survive completely. Their independent communities disappeared. Their traditions were not carried intact into the present. Their languages, stories, and understanding of the world were lost. What survived were fragments. Some fragments entered immune systems. Others influenced skin biology, oxygen response, pain sensitivity, or the timing of biological rhythms. Many produced effects too small to notice. Most disappeared entirely. Genetic continuation is therefore not the same as cultural survival, yet it still changes the way human history should be understood. Homo sapiens did not simply replace every other human population without being changed. Our ancestors entered new regions, encountered people who had lived there long before them, and absorbed small parts of their biology. The modern human genome became a record of migration, separation, reunion, and loss. This history also explains why ancient variants should not be described as turning against us. Genes do not contain intentions. A variant that once improved survival under frequent infection, cold, hunger, or injury does not become malicious when conditions change. The immune system still needs inflammation. The body still needs to store energy. Blood still needs to clot. Pain still needs to warn us about injury. These systems remain essential. Problems arise when the intensity, timing, or duration of a response no longer matches the environment. An immune alarm may remain active after the threat is gone. Energy storage may continue in a world of constant food availability. A clotting system shaped by physical danger may operate during decades of inactivity. Pain may continue after damaged tissue has healed. The biology is not useless. The balance has shifted. Evolution cannot predict what the future will demand. It does not build perfect organisms. It preserves traits that work well enough under existing conditions, even when those same traits carry disadvantages. Every adaptation has limits. Every advantage depends on context. The ancient fragments inside modern humans are examples of this rule. Some appear to have helped our ancestors survive Eurasian pathogens. Some help people tolerate high altitude. Some may have improved protection against environmental stress. Others were removed because they were incompatible or harmful. The fragments that remain are not trophies and not defects. They are evidence of survival under conditions that no longer exist in the same form. The people who first carried them are gone, but not everything they passed forward disappeared with them. A response to low oxygen still operates on the Tibetan plateau. Ancient immune variants still influence how some bodies react to infection. Small pieces of Neanderthal and Denisovan ancestry continue moving from parent to child, usually unnoticed. They survive not as separate peoples, but as biological echoes inside another human population. Our species is not the product of purity. It is the product of movement, contact, adaptation, and mixture. Homo sapiens survived partly because of culture, cooperation, and invention. But our ancestors also inherited solutions from humans who had already endured the cold, pathogens, mountains, and seasons of Eurasia. Those inherited solutions came with benefits. Some also came with costs. The world changed. The genes remained. They survived in us, and so did the price.